One particularly pathogenic agent that infects our body, more specifically our immune system, is a type of virus known as the human immunodeficiency virus, or simply HIV. Now, transmitted through bodily fluids such as blood and semen, what this virus does is it infects special white blood cells, special lymphocytes of our immune system, and transforms these healthy lymphocytes into virus-producing cells that ultimately lose all their functionality as immune cells. They lose their ability to defend our body from infections and from invading pathogens, and this greatly weakens our immune system and leads to a medical condition known as acquired immune deficiency syndrome, or simply AIDS. Now, AIDS is not the same thing as HIV. HIV is that pathogenic agent that eventually leads to AIDS. So this is the cause and this is the effect. So don't use these two terms interchangeably. They are not the same exact thing, but they are related. One causes the other. Now, let's discuss the mechanism by which HIV actually invades the white blood cells of our body. And let's begin by examining the structure of an HIV agent. So this is what HIV looks like. We basically have this membrane shown in pink, and on the membrane we have these embedded glycoproteins. Now, we have different types of glycoproteins. The two glycoproteins we're going to focus on in this lecture is a glycoprotein called GP120 and glycoprotein called GP41. So GP120 is positioned on top of GP41. Now, GP120 is actually used to bind onto receptors on the target cell's membrane, and GP41 is used to stimulate the process of fusion of the membrane of the virus and the membrane of the target cell. Now, inside this membrane, we have the protein capsid shown in brown, and inside that protein capsid, we have the contents of that virus. We have the genetic information, the RNA, shown in red, and we have two very important types of protein enzymes, one known as integrase. Let's suppose that's the blue one, that's the green one, and the other one is known as reverse transcriptase. Let's suppose that is our blue one, and we'll discuss what their function is in just a moment. So basically, the GP120 glycoprotein of HIV binds to a specific glycoprotein receptor found on membrane of immune cells. And this special type of glycoprotein is called CD4 glycoprotein. So HIV contains a glycoprotein, the GP120, that can bind onto the CD4 protein of immune cells, our white blood cells. Now, recall from our discussion on white blood cells and specifically on T lymphocytes, we said that one of the specialized types of T lymphocytes that contains a CD4 glycoprotein are the helper T cells of our adaptive, our acquired immune system. So recall that helper T cells are CD4 positive cells. And what that means is these HIV agents will be able to bind and will be able to attack these helper T cells. So these are the helper T cells that HIV actually attacks. So let's take a look at the following diagram. We have the HIV agent that uses the GP120 glycoprotein to bind onto the CD4 glycoprotein of this healthy helper T cell. Now, inside the helper T cell, we have these different types of organelles. So we only show the nucleus and we show our endoplasmic reticulum. This is the rough endoplasmic reticulum that synthesizes our proteins, and we have the genome, the DNA of that helper T cell inside the nucleus. Now, once this binding process takes place, what happens next is the GP41 glycoprotein, the other glycoprotein attached to the GP120 and to the membrane of our virus initiates or stimulates the cell-mediated endocytotic process. This is the process by which the membrane of the virus fuses with the membrane of the cell. And that essentially dumps all the viral contents into the cytoplasm of our cell, as shown in this diagram. So we have the fusion of the membrane in pink of the virus and the membrane in blue of that helper T cell. And so this is what happens. We have the blue section and we have the fused pink section. And the protein capsid along with the contents are now found inside the cytoplasm of our cell. So what happens next? Well, HIV is a type of virus known as a retrovirus. Now, what exactly is a retrovirus? Well, a retrovirus is a virus that contains RNA molecules as well as a special enzyme that catalyzes the formation of viral DNA from that viral RNA. So that's why we have reverse transcriptase, because it synthesizes our nucleotides in the opposite direction to what is normally seen in nature. Normally, we synthesize DNA to RNA, but in this case, it goes from RNA to DNA in the opposite process. So HIV is a retrovirus, which means it has an enzyme called reverse transcriptase that can form viral DNA from the viral RNA. So these blue molecules, once inside the cytoplasm, attached to the red RNA molecules, and they begin to synthesize the viral DNA. Now, once the viral DNA is synthesized, it moves into the nucleus of that helper T cell, and an enzyme known as integrase, shown in green, basically binds onto that DNA of that cell. It clips it, it opens it at a certain position, and it incorporates that viral DNA into that original genome, original DNA of the helper T cell. And now this is an infected helper T cell. So notice in this case, this was purely brown, but now a small piece of that viral DNA shown in red has been integrated with that genome of our helper T cell. Now that we have incorporated the viral DNA into our DNA of the cell, when that DNA is used to synthesize mRNA, we're going to synthesize viral mRNA to produce viral proteins. And in this manner, we transform our helper T cell into a factory that produces viral proteins and viral RNA molecules that ultimately causes the cell to lyse. And when it lyses, when it breaks open, it releases many HIV agents that go on to essentially infect other helper T cells of our body. And in this method, what HIV does is it essentially destroys our population of helper T cells in our body, and that weakens our immune system and leads to the acquired immune deficiency syndrome. And because we have the acquired immune deficiency syndrome, we have a very weak immune system. Any type of infection that enters our body can essentially kill us off, and that includes the common cold. So it's not the actual HIV, that virus, that kills us, but it's what it does that ultimately kills us. It's the fact that we create a very weak, or the virus creates a very weak immune system, and any infection whatsoever can essentially kill us off. Now, the next question is, what's the big deal about these helper T cells? Why is it that when these helper T cells are destroyed, our immune system becomes so weak? So we have many different types of white blood cells in our body, and helper T cells are only one of these different types of white blood cells. So what exactly makes the helper T cell so special? Well, it turns out that helper T cells are the cells that oversee all the different types of processes that take place inside our immune system. And they essentially allow that immune system to function effectively and efficiently. And although they do not attack pathogens directly, what they do is they bind onto T lymphocytes and they induce the differentiation of these T lymphocytes into other specialized white blood cells, such as cytotoxic T cells, that are important in fighting off infections and infected cells. They also bind onto B lymphocytes, and that induces the differentiation of these B cells into plasma cells that are needed to produce antibodies floating in our blood, and they also induce differentiation into memory B cells that store the antibodies in case we're ever reinfected with that same pathogen. Now, these helper T cells also release special chemicals called cytokines. And these cytokines not only induce white blood cell differentiation, but they also stimulate the cells of our innate immune system. These are our macrophages, neutrophils, basophils, and other white blood cells of our innate immune system. So we see that by destroying the population of our helper T cells, what HIV actually does is it severely weakens not only our adaptive, our acquired immune system, but also our innate immune system. And as a result, most individuals infected with HIV die not from the virus itself, but from other infections that infect our bodies, such as the common cold, pneumonia, the flu, or even cancer. And because our body cannot actually defend itself because our immune system is weakened, that is why we actually die. So we die because of our body's inability to mount a proper immune response as a result of the acquired immune deficiency syndrome, or AIDS. Now, the final thing I'd like to briefly discuss is the stages that the person goes through when they are infected with HIV. So there are 3 stages. We have the early stage, we have the middle stage, and we also have the late stage, which is the stage when the person is said to have AIDS. So early stage lasts about 2 weeks, and this is when the person actually feels sick. This is when the person experiences flu-like symptoms. For example, they have a high fever, they have different types of body aches, they have headaches, and this is when inside the blood there's a high level of these viral agents, the HIV agent. So basically the individual experiences flu-like symptoms, fever, aches, and so forth, and has a high level of virulence in the body. Now, the middle stage is the stage when the person doesn't actually experience any symptoms. So you have no way of actually knowing whether or not you are carrying the virus. The only way that you can know is if you go get tested. And that's because this is the stage when the body begins to produce antibodies. So we have antibodies floating in our blood that are specific to this virus. And these antibodies can be used to detect that virus in our body. So middle stage can last from several months to several years. So no visible symptoms, low levels of that virus, we have the presence of these antibodies, and we have a very slow but very continual decline in the population of our helper T cells. So during the middle stage, the HIV continually but slowly begins to kill off these helper T cells. And the final stage, the late stage, this is when we actually are diagnosed with acquired immune deficiency syndrome. This is when our HIV begins to kill off the helper T cells at a very high rate. And the population of helper T cells drops to a very low amount. And what that means this is when our innate and acquired immune systems are weakened to the point where if any infection takes place, such as the common cold, flu, pneumonia, we essentially will die because of our body's inability to mount some type of defensive response. And this is when the person can also develop cancer, and that's because our immune system loses its ability to fight off cancerous cells. Because if we can't produce cytotoxic T cells, and because if we can't release cytokines that influence natural killer cells, we don't have any cells that can destroy our cancer cells of the body, and that's why we ultimately develop cancer when we are at the late stage, when we have the acquired immune deficiency syndrome.